So this final set of slides deals with orchestration of the immune response. And what we want to do here is to take all of the pieces and all of the components that we've been talking about throughout the entire semester and try to fit them all together to see how they actually function together as a whole to help eliminate infection and provide immunity. And I'd like to start off by looking at one of the first slides that we showed you in one of the first lectures that we did, where we laid out the components of the immune system. We talked about innate immunity and adaptive immunity. And just to do a quick reminder that we were looking at physical barriers in the innate immune system, such as skin and mucous membranes. We were looking at chemical factors, and we are also looking at cellular defenses, such as neutrophils and basophils and eosinophils. We also looked at adaptive immunity, and in particular the B cell responses, which lead to, of course, antibody production, and the T cell responses, or the cell-mediated immunity, which result in elimination of host cells. We also talked a little bit about inflammation as being a process that spans both innate and adaptive immunity and is very powerful to promote defense against invading microorganisms. And we looked as well at cytokines and the role they play in signaling. So let's now see how these different components work together in the case of an actual infection. So we're going to put it all together, and there's a couple of things that you need to keep in mind. One of them is that all of these components work together. They all have a role to play, but not all components of the immune system are involved in every infection. So depending on the nature of the infectious microorganism, whether it's a bacteria or a virus, and in particular, depending on where the components, where the location of the infection is, a lot of times will decide what components come into play. And if we wanted to create two broad scenarios, we could talk about infections that occur outside of the cells. So let's say you have multiplication of bacteria outside of a host cell. Perhaps the bacteria are somehow attached to the outside of the cell, and they are causing illness, but they don't actually get inside the cells. We can also look at the case where you have perhaps maybe a virus particle that begins to infect and replicate inside the cell. So you have external pathogens and you have internal pathogens such as viruses. And we'll see that the immune system has a couple of different responses to each of these situations. So let's look first at the case of the external pathogens. So in this example, you can think of maybe perhaps an infection in the skin that's caused maybe by a splinter getting into the finger that becomes infected. So what is the immune system going to do in this case? Right? So one of the first things that happens at the site of infection, so inside the infected tissues, is that you get activation of the complement proteins in the bloodstream at that area. And remember, this is one of the first things that happens, so antibodies are not present at this time, which means that this has to be activation through the alternative pathway. And the alternative pathway, remember, does not need antibodies to activate complement. So these proteins that are normally in the blood and in an active state, they begin to become activated, as we talked about last time. And we know that some of the outcomes of complement activation are the production of this membrane attack complex, or MAC, which causes lysis of the bacterial cells. So already we are starting to try and destroy some of the infecting bacteria through these complement proteins. And we also get these byproducts produced, C3A, C5A, and C3b. And we'll talk in a second about what each of these factors would do. So complement activation, one of the very first things that happens. Now C3A and C5A complement proteins are able to bind to mast cells and release chemicals that trigger inflammation. So remember, mast cells are large cells that are in the tissues that are full of granules that contain these inflammatory chemicals. And one of them in particular is histamine. These inflammatory chemicals promote inflammation, and if you remember again back to the first lecture, inflammation causes the small blood vessels, the capillaries, to dilate or to expand, and that causes fluid and other chemicals to leak out, and it also allows neutrophils, it recruits neutrophils to the site of infection. Remember, they stick to the walls of the blood cells, squeeze their way out of the blood vessels, and they enter the tissues. So in this process of inflammation, you're getting neutrophils leaving the tissues, leaving the blood and entering the tissues. And this happens at the site of infection. Okay, so this initial inflammation is triggered by C3A and C5A. Now remember, neutrophils are phagocytic cells, and they're going to do most of the work to try and eliminate the microbes by phagocytosis. So most of the elimination is being done by the neutrophils. At the same time, within the tissues already are macrophages. Remember, macrophages reside in the tissues. So the macrophages in the tissues are also engulfing the microorganisms by phagocytosis. And in the case of both the neutrophils and both the macrophages, the complement protein C3b that's been produced is going to assist this process by acting as an opsonin. Remember, opsonins are factors that coat the bacterial cell and also bind to the phagocytic cell so they can bring the two cells together. The thing with macrophages that makes them different from neutrophils is that not only are they eliminating the cells by phagocytosis, but in the process they're becoming activated. And when they become activated, they secrete cytokines and other inflammatory molecules. So the macrophages act as what we call sentinels. And the job of a sentinel or a guard or somebody keeping watch is to alert the immune system that an infection is taking place and cause the other components of the immune system to become activated or to get ready to become activated. And they do that through secreting these cytokines and other inflammatory mediators. So when a macrophage engulfs pathogen, it's going to promote inflammation. Inflammation in turn is also going to stimulate the macrophage, which causes it to promote more inflammation, and the inflammation causes more stimulation of the macrophage. So you get this positive feedback loop. And what this does is it allows the inflammatory response to go from a very low level to a very high level very quickly. Okay, so now the immune system's becoming primed to deal with the infection. Okay, all of this up to this point is taking place in essentially the innate immune response. Now at the same time, you are also getting phagocytosis of the antigens by antigen-presenting cells such as dendritic cells and also macrophages. And their job, in this case, the bacteria are being processed and placed onto the surface of these antigen-presenting cells. And you should know by now that it's in association with MHC class II plus the antigen fragment. And these cells are then being carried to the secondary lymphoid tissues where they're going to present these antigens to B cells and T cells, present them to T cells, which in turn will activate the B cells. Okay, so the antigen presenting cells in the tissues encounter the antigens, process them, travel to the secondary lymphoid tissues to carry out antigen presentation. Now, some of the antigen also might make its way to the lymph nodes and encounter the antigen presenting cells there as well. And as we talked about over the last couple of weeks, the antigen presenting cells are then going to start the process of adaptive immunity, where you get activation of the Th1 and Th2 helper T cells, which are going to be helping to activate the B cells, these guys in particular. So the B cells become activated, the B cells secrete cytokines, they can travel to amplify the inflammatory response, and most importantly, this leads to production of antibodies. And we talked last time about what those antibodies might do, and in particular, you're looking at more complement activation. So you just keep the process, the inflammatory response continuing. And you're also looking at opsonization, neutralization, complex formation, so immune complex formation. All of these things enhanced by the antibodies. So as you can see, the adaptive response actually comes into play quite late in the game, and it's the innate responses which are doing most of the heavy lifting towards the beginning. Now, in the case of intracellular microbes, so these are things like viruses that are replicating inside the cell, you have to remember that before the virus gets into the cell, it's going to spend a little bit of time outside of the cell. So before the virus has a chance to infect the cell, you are going to get antigen processing and presentation by antigen presenting cells, just as you would with the inflammatory response. You're going to get activation of Th1 cells, which in turn is going to lead to activation of the cytotoxic T cells. So everything that we talked about in the previous example, the inflammatory response, complement activation, neutrophils, they also will play some role, but the key difference here is that you are going to be activating Th1 cells by the intracellular pathogens, which in turn are going to activate the cytotoxic T cells. So this is the big difference. The cytotoxic T cells really only come into play when you're dealing with infected host cells, and they don't necessarily have a role to play if you're dealing with an extracellular microbe such as a bacterial infection. Other than that, the inflammatory response is basically very, very similar. So viruses and intracellular microbes will engage the cytotoxic T cells, whereas the external bacterial infections will not. Okay. You also have natural killer cells, which may become involved by the antibodies that are being produced by the B cells, and the whole process of ADCC, or antibody-dependent cell-mediated cytotoxicity. So these are the two key differences between the intracellular pathogen response and the extracellular pathogen response. All right, so that concludes the lectures for this course. I hope you enjoyed it, and good luck on your final exams.